In our everyday lives, we get exposed to different situations wherein trillions of microbes found in the air, the food we eat, and the objects we touch are trying to invade our body. But have you ever wondered why we're healthy most of the time? This is because of the immune system. We may be unaware, but inside our body, our immune system works very hard in fighting with the viruses and bacteria that are trying to kill our cells. In this video, we are going to explain to you about how all of these happen. Let us start by knowing what the immune system is made up of. Here are the organs that comprise the immune system. These are the tonsils. The tonsils are often the first organs to encounter pathogens and antigens that come into the body by mouth or by nose. There are three pairs of tonsils in a ring about the pharynx. The pharyngeal tonsils, which reside behind the nose, the palatine tonsils, which reside on both sides of the back of the throat, and the lingual tonsils, which reside at the back of the tongue. These are the lymph nodes. The lymph nodes are small oval-shaped structures located along the lymphatic vessels. They are about 1 to 25 millimeters in diameter. Lymph nodes act as filters with an internal honeycomb of connective tissue, filled with lymphocytes that collect and destroy bacteria and viruses. They are divided into compartments, each packed with B lymphocytes and a sinus. As lymph flows through the sinuses, it is filtered by macrophages. Lymph nodes are in each cavity of the body except the dorsal cavity. Physicians can often detect the body's reaction to infection by feeling for swollen, tender lymph nodes under the armpits and in the neck, because when the body is fighting an infection, these lymphocytes multiply rapidly and produce a characteristic swelling of the lymph nodes. This is the thymus gland. The thymus gland is located in the upper thoracic cavity, posterior to the sternum and anterior to the ascending aorta. The thymus is an organ that is more active in children and shrinks as we get older. Connective tissues separate the thymus into lobules, which contain lymphocytes. Thymic hormones such as thymosin are produced in the thymus gland. Thymosin is thought to aid in the maturation of T lymphocytes. The thymus is critical to the immune system. Without a thymus, a person has no ability to reject foreign substances. Blood lymphocyte level is very poor, and the body's response to most antigens is either absent or very weak. These are the Peyer's patches. The Peyer's patches are located in the wall of the intestine and in the appendix. They are attached to the cecum of the large intestine. They intercept pathogens that come to the body through the intestinal tract. This is the spleen. The spleen is a ductless, vertebrate gland that is closely associated with the circulatory system, where it functions in the destruction of all red blood cells and holding a reservoir of blood. Located in the upper left region of the abdominal cavity, it is divided into partial compartments. Each compartment contains tissues known as the white pulp and the red pulp. The white pulp contains lymphocytes and the red pulp acts in blood filtration. When blood enters the spleen and flows through the sinuses for filtration, lymphocytes react to pathogens. Macrophages engulf debris and also remove old, worn-out red blood cells. A person without a spleen is more susceptible to infections and may need supplementary antibiotic therapy for the rest of their life. This is the red bone marrow. The red bone marrow is a soft, spongy, nutrient-rich tissue in the cavities of certain long bones. It is the organ that is the site of blood cell production. Some of the white blood cells produced in the bone marrow are neutrophils, basophils, eosinophils, monocytes, and lymphocytes. Red bone marrow is also the site of maturation of the B lymphocytes, while T lymphocytes mature in the thymus. The immune system also needs cells. Here are some of the cells that comprise the immune system. The macrophages. Macrophages are the primary scavengers within tissues. Macrophages also remove larger particles, such as old RBC and dead neutrophils. Macrophages play an important role in the development of acquired immunity. After they ingest and digest molecular or cellular antigens, fragments of processed antigen are inserted into the macrophage membrane as part of the surface protein complexes. The eosinophils. Eosinophils fight parasites and contribute to allergic reactions. They are easily recognized by the bright pink staining granules in their cytoplasm. Normally, there are only a few eosinophils found in the peripheral circulatory. They account for only 1 to 3% of all leukocytes. The lifespan of a typical eosinophil in the blood is about 6 to 12 hours. Eosinophils are known to attach to large parasites and release substances from their granules that damage or kill the parasite. Because eosinophils kill pathogens, they are classified as cytotoxic cells. Eosinophils also participate in allergic reactions by contributing to inflammation and tissue damage by releasing toxic enzymes. The basophils. Basophils release histamine and other chemicals. They are rare in circulation but are easily recognized in a stained blood smear by the large, dark blue granules in their cytoplasm. They also release mediators that contribute to inflammation. The granules contain histamine, heparin, cytokines, and other chemicals involved in allergic and immune responses. The monocytes. Monocytes are the precursor cells of tissue macrophages. Monocytes are not that common in the blood. Once out of blood, monocytes enlarge and differentiate into macrophages. Some tissue macrophages patrol the tissues, creeping along the amoeboid motion. Others find a location and remain fixed in place. The T lymphocytes. T lymphocytes are a type of lymphocyte that play a certain role in the cell-mediated immunity. They can be distinguished from other lymphocytes, such as B lymphocytes and natural killer cells, by the presence of a T cell receptor or TCR on the cell surface. They do not have antigen-presenting properties, rather they require B lymphocytes or natural killer cells for its antigen-presenting property. They are called T cells because they mature in the thymus. There are several subsets of T cells, such as helper, cytotoxic, memory, and regulatory T cells, each with a distinct function. The B lymphocytes. B lymphocytes, like the T lymphocytes, play important roles in cell-mediated immunity. They can be distinguished from other lymphocytes by the presence of a protein on the B lymphocyte's outer surface known as the B cell receptor or the BCR. This specialized receptor protein allows a B lymphocyte to bind to a specific antigen. The principal functions of B lymphocytes are to make antibodies against antigens, to perform the role of antigen-presenting cells, and to develop into memory B cells after activation by antigen interaction. They are called B lymphocytes because they form at the bone marrow. The natural killer cells. The natural killer cells have antigen-recognizing receptors, like the B and T lymphocytes on their surface. They attack foreign cells or abnormal cells, like cancer cells and virus-infected cells, directly without any previous stimulation. The dendritic cells. Dendritic cells activate lymphocytes. They are antigen-presenting cells characterized by long, thin processes that resemble neuronal dendrites. Dendritic cells are found in the skin called Langerhans cells and also in various organs. When dendritic cells recognize and capture antigens, they migrate to secondary lymphoid tissues, where they present the antigens to lymphocytes.
The effector phase begins when activated cytotoxic T cells, which were stimulated to proliferate by the cytokine IL-2, recognizes the MH class I antigenic peptide complex on the infected epithelial cells. The cellular immune response. The activation phase begins when an antigen-presenting cell of the host organism encounters and attacks an invading virus. Meanwhile, other viruses look for nearby cells such as epithelial cells to infect. A lysosome containing digestive enzymes combines with the phagosome to process antigens. The processed antigens combine with the MHC class II proteins and are presented on the surface of the APC. The virus also infects the epithelial cells. Within the infected epithelial cell, the virus is processed, attached to an MHC class I protein, and is presented on the cell surface. A helper T cell recognizes the displayed antigen on the APC and binds to the MHC class II antigenic peptide complex. The activated helper T cell releases chemical messengers such as cytokine IL-2 and gamma interferon. Cytokines also attract killer T cells to the site of infection. The activated cytotoxic T cell binds to the MHC class I antigenic peptide complex on the surface of the infected epithelial cells. The binding causes the cytotoxic T cell to release a potent chemical called perforin. Perforin perforates the cell membrane of the infected cells, causing the cells to lyse and die. As the viral infection is brought under control, the activated cytotoxic T cells are turned off by regulatory T cells. Memory T cells remain behind to respond quickly if the same virus attacks again. The humoral immune response. The activation phase begins when an invading bacteria is phagocytized by an antigen-presenting cell. A lysosome containing digestive enzymes combines with the phagosome to process the antigens. The processed antigens combine with the MHC class II proteins and are presented on the surface of the APC. Helper T cells recognize the displayed antigen on the APC and bind to the MHC class II antigenic peptide complex. This binding triggers the APC to release the cytokine IL-1, which activates the helper T cell. The activated helper T cell releases the cytokine IL-2, which stimulates the T cell to proliferate. Thus producing many T helper T cells, each with a receptor specific for the original process antigen. The effector phase begins when a B cell that exhibits on its surface an LGM receptor specific for the same antigen, originally engulfed by the APC, encounters and binds the antigen. The B cell engulfs the complex by receptor-mediated endocytosis. The phagosome containing the antigen fuses with a lysosome. The antigen is processed. The processed antigen binds MHC class II protein and is displayed on the surface of the B cell. Helper T cells now bind to the displayed antigen on the surface of the B cell, causing the helper T cell to release cytokines. The cytokines stimulate the B cell to divide and proliferate into identical B cell copies. The B cells differentiate into antibody-producing plasma cells and memory cells. The plasma cells release antibodies with a specificity identical to that of a surface receptors on the parent B cell. These carry out the ultimate goal of fighting the foreign invaders. The released antibodies bind in a lock and key fashion to the antigen on the surface of the original bacterial invaders. This makes it easier for killer cells to attack and destroy the bacteria by phagocytosis, and the release of proteins causes the direct lysis of the bacteria. Simultaneously, a blood component known as complement is signaled to attack and puncture holes in the bacteria.
Lymphocytes, or white blood cells, form in the bone marrow and active in lymphatic tissues. Lymphocytes are one of the 5 kinds of white blood cells or leukocytes in the blood. B lymphocytes are often called as B cells, mature in the bone marrow, while T lymphocytes, or more commonly known as T cells, mature in the thymus gland. There are different kinds of T cells: helper T cells, killer T cells, suppressor T cells, and memory T cells. What do T cells and B cells do? T cells and B cells are highly specialized defender cells. Different group of cells are tailored to different germs. When your body is infected with a particular germ, only the T cells and B cells that recognize it will respond. These selected cells then quickly multiply, creating an army of identical cells to fight the infection. Special types of T cells and B cells remember the invader, making you immune to a second attack. How do you recognize invaders? Your T and B cells recognize invaders by the shape of the molecules, antigens, on their surfaces. Your immune system can produce a T and B cell to fit every possible shape. However, any T or B cells that recognize molecules found on your cell were destroyed while you were growing the womb to prevent them from attacking your own body, but you were left with millions of others, one for every foreign antigen you might ever encounter. What's so special about your T cells? Having recognized the invader, different types of T cells then have different jobs to do. Some send chemical instructions to the rest of the immune system. Your body can then produce the most effective weapons against the invaders, which may be bacteria, viruses, or parasites. Other types of T cells recognize and kill virus-infected cells directly. Some help B cells to make antibodies which circulate and bind to antigens. What's so special about your B cells? With the help of T cells, B cells make special Y-shaped proteins called antibodies. Antibodies stick to antigen on the surface of the germs, stopping them in their tracks, creating clumps that alert your body to the presence of intruders. Your body then starts to make toxic substances to fight them. Patrolling defender cells called phagocytes engulf and destroy antibody-covered intruders. More than 2,000 years ago, the Greek historian Thucydides recognized that occasionally someone contracts a disease, recovers, and never catches that particular disease again. The person becomes immune. With rare exceptions, however, the immunity to one disease confers no protection against other diseases. Thus, the immune system attacks one type of microbe, overcomes it, and provides future protection against that microbe, but no other. This is why we refer to the immune response as specific defense against invasion. The immune system consists of about 2 trillion specialized white blood cells called lymphocytes. Many are distributed throughout the body in the blood and lymph. Others are clustered in specific organs, particularly the thymus, lymph node, and spleen. The immune response results from the interaction of among several types of lymphocytes and the molecules they produce. The immune response is provided by two types of lymphocytes, B cells and T cells. As with all the white blood cells, B and T lymphocytes arise from cells in the bone marrow early in development. Newly forming T cells migrate to the thymus, hence the name T cells, and differentiate. B cells differentiate in the bone marrow itself, hence the name B cells, and later migrate typically either to the spleen or lymph nodes. Immune responses produced by both T cells and B cells composed of 3 steps: recognizing invaders, launching their attacks, and retaining the memory of their invaders to ward off future infections. To understand how the immune system responds to invading microbes, 3 questions must be answered. How do immune cells recognize foreign molecules? How do they produce specific responses to many different molecules? And how do they determine that the substance is foreign and not self, thereby avoiding disruption of the wrong body tissues? The key to the immune system's ability to attack invading microbes lies largely with proteins called antibodies and T cell receptors. Antibodies are either attached to the surfaces of the B cells or dissolved in the blood plasma where they are called immunoglobulins. T cell receptors are only found attached to the surfaces of T cells. Antibodies are Y-shaped molecules composed of 4 peptide chains, one large heavy chain and one small light chain on each side of the Y. Both heavy and light chains consist of a constant region that is similar to antibodies of the same class, and a variable region that differs among antibodies. The combination of light and heavy chains results to an antibody with 2 functional parts, the arm and the stem of the Y. The variable region of the arms form highly specific binding sites for large molecules. Each binding site has a particular shape and an electrical charge, so only a few or perhaps only one type of molecule can fit in and bind to the site. In general, only large complex molecules such as proteins and polysaccharides bind to antibodies. These molecules or antigens may either be attached to the surfaces of the cells, the body's own or the microbes, or dissolve in the blood or extracellular fluid such as snake venom. We will discuss shortly the binding of antigen to antibody triggers the immune response. The stem of antibody molecules, which consists of the part of the constant regions of 2 heavy chains, determines the activity of the antibody. Though the constant regions and different antibodies are similar, they are not identical. For example, the heavy chain's constant regions forming the stem of one type of antibody may attach to the antibody to the plasma membrane of the cell, but constant regions of another type of an antibody may bind to a certain protein in the blood called complement to promote the destruction of microbes.
Lymphocytes are a type of white blood cell. And so basically there are two types of lymphocytes. There are B lymphocytes and then there are T lymphocytes, which we'll get to in just a second. Okay, so B lymphocytes are made in the bone marrow and they're responsible for a humoral response. Now this term I'm going to show you a couple more times in this podcast. Humoral means in the fluid or in the humors of your body. So that means in the blood, in the lymph material, in the lymph vessels, in the interstitial fluid. And so humoral response, what I want you to think about, is going to be anytime there are viruses free in the fluids of our body. And what the B lymphocytes do is they produce antibodies. So how does that work? Basically, you have a naive B cell. It's going to sense the shape of the antigen. I'll tell you how that works in just a second. But basically what it's going to do is it's going to produce antibodies. And so what B lymphocytes do is they produce all of these antibodies that are specific for the antigen. So in other words, if we're infected by this virus, we'll call this virus A1, then we're going to produce antibodies for that specific antigen. We're also going to produce memory B cells so that we have that immunity for the rest of our life. Well, the T lymphocytes are responsible for cell-mediated response. What does that mean? They're going to target and kill the cells inside our body that are already infected. Okay, so what are the T lymphocytes? Where are they made? They're made in the thymus gland, which kind of sits on top of our heart. Basically what they do is they create what are called killer T cells or killer T lymphocytes. And so a killer T cell is an activated T cell. Basically what it'll do is it'll find any of the cells inside our body that are infected with a virus, it'll dock next to them, and it will kill our own cells. And so it's going to kill any of the cells inside our body that are infected by the virus, or even cancerous cells, it's going to kill them. And so it's made in the thymus, and it produces cell death inside us. And so the T lymphocytes are responsible for the cell-mediated, killing cells inside our cells that are infected. B lymphocytes are going to be in the humors of our body.
Allergens are usually harmless foreign substances that trigger the body's immune system to react by producing the antibody immunoglobulin E or IgE in abnormal quantities. Common allergen includes dust mites, pet dander, food, insect venom, and pollen. When people who have allergies encounter allergen, such as pollen, for the first time, their immune system mistakenly believes that the allergen is a threat and responds by producing antibodies to fight them. These antibodies bind to specialized cells called mast cells. Once the body is exposed to the same allergen, it binds to the antibodies on the surface of the mast cells and triggers the release of the chemicals such as histamine and leukotriene. The release of these chemicals causes the symptoms associated with allergy. Symptoms may include itchy and watery eyes, nasal congestion, sinus pressure, runny nose, sneezing, shortness of breath, chest tightness, coughing, and hives. Allergies can affect anyone, regardless of age, gender, race, or status in life. However, a first-time occurrence can happen at any age or recur after many years of remission. Hormones, stress, smoke, perfume, or environmental irritants may also play a role in the development of severity of allergies. There are no cure for allergies, but there are ways to relieve or prevent allergy symptoms. These include avoidance of substances to which you're allergic, use of medications, and in certain cases, immunotherapy or allergy shots.
What is HIV and AIDS? AIDS stands for Acquired Immune Deficiency Syndrome, a complication derived from a viral infection called Human Immunodeficiency Virus, often called HIV. HIV viruses generally live on semen, vaginal secretion, and blood and breast milk. The virus could usually be transmitted by sexual intercourse, breastfeeding, and the sharing of drug paraphernalia. HIV could rarely be transmitted through blood transmission, and remember that HIV virus can be transmitted through saliva but through oral sex. An HIV virus mainly targets the CD4 cells or T cells, a part of the human body's immune system, making them weak. Upon contact, the HIV virus makes itself a part of the DNA or the genetic code, meaning upon creation of other T cells, HIV viruses are also produced. HIV virus first symptoms are flu-like, and yet these symptoms could disappear without any treatment. But upon creation of antibodies for flu, the HIV also continuously degrades our immune system, and signs could appear in several months up to 15 years. Untreated HIV will soon lead to AIDS. Taking note that the people with HIV doesn't mean they have AIDS, but all people with AIDS have HIV. AIDS is usually detected through saliva or blood tests that measures the number of CD4 cells in a person's body. If the T cell count drops below 200, this means our immune system is severely damaged, making the body incapable of defending the body from extremities. A person could also be diagnosed with AIDS if the person has one of the 26 opportunistic infections or conditions that are mostly found in advanced HIV patients, because these conditions would not occur if the human body immune system works efficiently. How to treat AIDS? For more than 30 years of HIV pandemic, we have done a lot of the people that could not imagine, but still, we still have a long way to go. In America, there are at least 1.2 million victims of HIV, and after all the efforts of the government, the number of HIV patients would increase by 50,000 annually. And the people living with HIV, only 82% knows that they are infected, 66% has the link to care, 37% would stay in care, 33% are in antiretroviral therapy, and only 25% would adhere to the treatment and has very low amount of virus in their blood. Seeing the statistics, it only shows that 3 out of 4 HIV patients would not get the proper treatment to manage the disease and keep the virus under control, and overview of 3/4 of a million. But what if we transform our response on the social and economic drivers of risk? What if we heighten the HIV prevention effort for people with positives and negatives? What if we improve systems that diagnose HIV and make them linked to care, help them stay in care, expand treatment with adhering support? As a result, fewer people would fall out of care and more people could lower their viral load, making them live healthier and longer. As a result, the chances of spreading the disease would be limited, and someday there would be an AIDS-free generation.